This is a video short to introduce genetics, and it'll be the first in a series of three videos that will introduce and go over how to use the Punnett squares, the basics of genetics. So, start out at the beginning. Should have the PowerPoint that can be printed from the website, or you should have a copy of it. Genetics, the field of study that studies how our traits are passed on to the next generation. And not just our traits, but all organisms, because the genetic code's pretty universal. You know, when you look at the DNA of us, DNA of an oak tree, DNA of a mosquito, they all code for different things, but they're using the same code. They're using the same type of code, DNA. From our last unit, you know that egg and sperm are gametes, and after meiosis, we reduce the number of chromosomes that are passed on to 1/2 of our set. So for each trait, we have two choices. These two choices are called alleles. And some of these words are in red up here on the screen. You might want to highlight them or mark them so that maybe you can make flashcards or something like that later on out of index cards. But each parent has two choices for a trait, and they can only pass on one, because remember, after meiosis we split those two choices up so that in an egg or a sperm there's only one choice for each trait. So we call those alleles. If a parent has both the same choices, like we're going to use letters, say capital T, that's called homozygous. Or little t, that's also homozygous. The term heterozygous means that the two choices are different. So like a big T, little t would be a heterozygous pair of alleles. So when we look at the parents, the actual letters they have, we have a name for that. We call it the genotype. So you can kind of think of it like gene type. Big T and little t are two different gene types. Big T would be the other possible choice of gene type or genotype. We're going to do a method called the Punnett square in a little bit to show how these are passed on to the next generation. And you'll be asked for the genotypic ratio. And it might be 2:2, or it might be 3:1, or 4:0. Sometimes it's stated in terms of percent, 75% tall, 25% short. That would be the phenotypic ratio. Phenotype means physical type. So think of physical and pheno. Phenotype, physical type. Tall, short, red, yellow, green, rough, smooth. Those are all phenotypes. So you'll notice that we'll talk about tall and short a lot. Gregor Mendel was the person who gave us, we call him the father of genetics. He gave us a lot of what we're going to talk about today as far as how to study genetics and how to represent it. He did most of his work with pea plants, and tall and short is one of those kind of those mainstay characteristics. Tall plant is dominant, short plant is recessive, and it is a simple dominance trait that, or complete dominance trait that we're going to talk about. So again, genotype would be the actual letters. Phenotype would be what they look like after. So we're going to talk about simple dominance first. There are a couple other types of dominance, incomplete and codominance, that we'll talk about later. But whenever there is a dominant trait and a recessive, we call this complete dominance or simple dominance. And in simple or complete dominance, there's a dominant allele and a recessive. If you have or possess the dominant allele, you show that trait. That's it. It doesn't matter what the other one is. So for pea plants, tall is dominant. Big T means you're tall pea plant. If you have big T, well, it's still tall. If it's big T, it's still tall. And in most cases, it's no taller than the heterozygous big T little t. It just means that if you have that, you're tall. The only way you can be short in pea plants is have little t, little t. Now, we keep talking about these letters. There is a little bit of a trick to that. Whenever you see a simple dominance problem, look for the dominant trait. That's the letter we're going to use for the trait. The capital represents the dominant trait, and the lowercase represents the recessive. And that's pretty standard. So always try to look for the dominant trait and use that letter for simple or complete dominance. So again, if a dominant allele is present, you're going to be that phenotype. You're going to look like whatever that dominant trait is. Recessive trait can only be shown if it's little t in this case, or if it's both recessive. That's the only time where that trait can show, is if there's no dominant trait to overshadow it. So again, tall and short pea plants, that's one of our standard traits that we're going to talk about. So we're going to look at Punnett squares. R.C. Punnett was a British mathematician and biologist who invented the Punnett square, and it's a way for us to represent the characteristics that we've been talking about. And we're going to start out with a four-square, which is just a single trait being passed on. Later on, we're going to get to a double trait, which has a bigger square. So let's look at it. This is what a Punnett square looks like, a little four-square. So you're going to make a box, and you're going to draw a line down the middle, and then down the middle horizontally. Notice that up at the top, we've got letters. This represents one of the parents, and this one represents the other. I think mostly the standard is that the male, if there's male and female, that the male is the parent represented on the top and the female on the side. But they're different. Different people look at it different way, but I think that's the standard if you can. It helps you know that if you do a problem and you're putting the wrong genotype there, then it helps me know that, well, you meant that to be the male and you messed up on that one. So we draw the little square, and then we put one of the alleles from the parent here, and we put one of the alleles, the other allele for the same parent here. On the other side, we put one of the alleles here and one here. And then what we want to do now is see what are the possibilities for passing this on. Remember, what we found out in last chapter is a parent can only pass on one of the alleles. So in this case, they're passing on the big T, and you would put a big T here and a big T here. Notice that there's a big T in both of the squares. Here, there's a little t, so we drop the little t here and the little t here. Then we go over to the side, and we take the big T and put a big T here and a big T here, and a little t here, and a little t here. That's all the possible ways that these parents can pass on the traits. In this particular case, we have one big T, two big T little t's, and one little t. Remember, if they're both the same, it's homozygous. So this one is homozygous. Recessive is the only short plant that is represented. So if there are four, there'd be one short plant and three tall. Or if there were many, many more, we could use this on a big scale and say 25% or 1/4 are going to be short and 75% are going to be tall. Now, there are two ratios we can get from this. We can get the genotypic ratio, which says what are the exact letters. So there's 1/4 or big T, 1/2 or big T, and 1/4 or little t. So our genotypic ratio is 1 to 2 to 1. The phenotypic ratio is the physical appearance. 3 out of 4 have tall. So the phenotypic ratio would be 3 to 1. Or 75% tall, 25% short. Genotypic and phenotypic ratio. Remember, these were two heterozygous parents, so we know they're big T, little t, and we showed the cross. This is how we take the parents and establish them onto the Punnett square, the big T, we took one of the alleles and put it in the first one. Usually the dominant is in the upper left corner, and if there's a recessive, then it will be on the right. So it's kind of a pattern that you try to follow as opposed to switching. Now, notice up here, we've got what we call the X format, the two parents, big T, little t, crossed with a big T, little t, two heterozygous tall parents. That's called the X format. It's a way that we could show it without a Punnett square. And you might be given this, and then you'll start and say, oh, well, this is how the Punnett square would look. One of the parents is here, and one of the parents is there. So we're going to do another Punnett square. We're going to represent the cross of a homozygous tall with a short plant. And you say, well, wait a minute, it's short. Well, how do we know what it's made of? Remember, it can only be short if it's homozygous little t, little t. So when we show this cross, you'll see that everything's the same. And why is that? Well, because all this can pass on. The male parent can only pass on a big T, and the other parent can only pass on a little t. So the only way you can get combinations would be big T, little t. So this is 4-0 or 100% tall or 100% big T, little t for the genotype. So you try it now. The ability to roll your tongue, believe it or not, is a trait that is said to be controlled by simple complete dominance. So see if you can roll your tongue. Look at maybe your lab partners. It is a dominant trait to be able to roll the tongue. Now remember, I want you to do the X format for this characteristic, and I want you to do a Punnett square, and then I want you to say what the genotypic ratio and the phenotypic ratio are at the end. And this is what we're going to show. You're going to show a cross of a heterozygous tongue-rolling parent with another heterozygous tongue-rolling parent. So you've got a little bit of time. You want to write on your maybe your note paper on the back or somewhere on your note paper. Work out the characteristics, or work out the X format and the Punnett square for this. And we'll take a little break and see how you do.